Cell division is a fundamental process that allows organisms to grow, repair damaged tissues, and reproduce. It is the method by which a single cell divides into two or more daughter cells. Growth is one of the primary reasons for cell division. As organisms grow, their cells must divide to increase in number. Additionally, cell division is crucial for repair. When tissues are damaged, cell division produces new cells that replace the old or damaged ones. Reproduction is another critical aspect of cell division. In multicellular organisms, specialized cells undergo division to produce gametes, sperm and eggs. Understanding cell division is pivotal in biology and medicine, laying the foundation for developments in genetic research, cancer treatment, and regenerative medicine. Mitosis and meiosis are the two primary types of cell division, each serving distinct purposes. Mitosis is the process by which a single cell divides to produce two genetically identical daughter cells. This type of division is crucial for growth, tissue repair, and asexual reproduction in multicellular organisms. Meiosis, on the other hand, is a specialized form of cell division that occurs in the production of gametes, sperm and egg cells. Unlike mitosis, meiosis results in 4 genetically distinct daughter cells, each with half the number of chromosomes of the parent cell. Meiosis introduces genetic variation through processes like crossing over and independent assortment. Mitosis ensures that each new cell is a carbon copy of the original, maintaining the organism's genetic integrity. Meiosis ensures genetic diversity among offspring, crucial for evolution and adaptation. Mitosis occurs in all somatic cells, while meiosis is confined to the reproductive organs. Understanding these processes provides a comprehensive view of how life proliferates and evolves. The cell cycle is a series of stages that a cell goes through to divide and produce new cells. It is composed of 4 main phases: G1, Gap1, S, synthesis, G2, Gap2, and M, mitosis. The G1 phase is the first stage of the cell cycle. During this phase, cells grow and carry out their normal functions. Next is the S phase, during which DNA replication occurs. Each chromosome is duplicated, resulting in 2 identical sister chromatids. Following the S phase is the G2 phase, where the cell continues to grow and prepare for mitosis. The final stage is the M phase, which includes mitosis and cytokinesis. Together, these processes produce 2 identical daughter cells, completing the cell cycle. Mitosis is a multi-step process that ensures the accurate division of a cell's genetic material. It consists of 4 main stages: prophase, metaphase, anaphase, and telophase. Prophase is the first stage of mitosis. During prophase, the chromatin condenses into visible chromosomes. The nuclear envelope begins to break down, and the mitotic spindle starts to form. Metaphase follows prophase and is characterized by the alignment of chromosomes along the metaphase plate. Anaphase is the next stage, where the sister chromatids are pulled apart by the spindle fibers. Each chromatid is drawn towards the opposite poles of the cell. Telophase is the final stage of mitosis. During telophase, the chromosomes reach the poles and begin to decondense, marking the end of nuclear division. Cytokinesis is the process that follows mitosis, involving the division of the cell's cytoplasm to form 2 distinct daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. This furrow forms as a contractile ring of actin and myosin filaments tightens around the cell's equator. The ring contracts progressively, pinching the cell membrane inward until the cell is split into 2 separate daughter cells. In plant cells, cytokinesis occurs differently due to the presence of a rigid cell wall. Instead of a cleavage furrow, plant cells form a cell plate at the center of the cell. Vesicles derived from the Golgi apparatus coalesce at the center, depositing cell wall materials to build a new cell wall. This plate gradually expands outward until it fuses with the existing cell membrane. Cytokinesis is the final step in cell division, culminating in the production of 2 new cells from a single parent cell. Meiosis is a specialized form of cell division that produces gametes, sperm and egg cells, with half the number of chromosomes of the parent cell. This reduction is crucial for maintaining the species' chromosome number across generations. Meiosis begins with prophase 1. where homologous chromosomes pair up and exchange genetic material through a process called crossing over. The homologous pairs then align at the metaphase plate during metaphase 1 and are separated to opposite poles in anaphase perverse. Telophase 1 and cytokinesis follow, resulting in 2 haploid cells. Meiosis II resembles a typical mitotic division. In prophase 2, the chromosomes condense again and the spindle apparatus forms. The chromosomes align at the metaphase plate during metaphase 2, and the sister chromatids are separated during anaphase 2. Telophase 2 and cytokinesis complete the process, resulting in 4 genetically distinct haploid cells. Genetic variation is a cornerstone of evolution, and meiosis plays a crucial role in generating this diversity. 2 key mechanisms during meiosis contribute to genetic variation-independent assortment and crossing over. Independent assortment occurs during metaphase 1 of meiosis. Homologous chromosomes align independently at the metaphase plate, resulting in a random distribution of maternal and paternal chromosomes to the daughter cells. Crossing over, which occurs during prophase 1, involves the exchange of genetic material between homologous chromosomes. This process creates new combinations of genes, further increasing genetic variability. The combined effects of independent assortment and crossing over ensure that each gamete, and consequently each offspring, is genetically unique. This genetic variation is vital for the survival and adaptation of species. Understanding these mechanisms offers insights into genetic disorders and potential therapeutic approaches. The regulation of cell division is a tightly controlled process involving various proteins and checkpoints. Cyclins and cyclin-dependent kinases, or CDKs, are the primary regulators of the cell cycle. These molecules ensure that cells only divide when conditions are favorable, and that each phase of the cell cycle is completed accurately. Cyclins are proteins whose levels fluctuate throughout the cell cycle. They bind to and activate CDKs, which are enzymes that phosphorylate target proteins to drive the cell cycle forward. Different cyclin CDK complexes are active at various stages of the cell cycle, ensuring precise control of cell division. Checkpoints are critical control mechanisms that monitor and regulate the progression of the cell cycle. The G1 checkpoint ensures that the cell is ready for DNA synthesis. The G2 checkpoint verifies that DNA replication is complete and accurate, and the M checkpoint ensures that all chromosomes are properly aligned before division. The interplay between cyclins, CDKs, and checkpoints ensures the fidelity of cell division. Deregulation of these processes can lead to uncontrolled cell division, contributing to diseases such as cancer. Cancer is fundamentally a disease of uncontrolled cell division. It arises when the regulatory mechanisms that control the cell cycle are disrupted, leading to excessive cell proliferation. This deregulation can result from mutations in genes that encode for cell cycle regulators, such as cyclins, CDKs, and checkpoint proteins. One common cause of cancer is the mutation of tumor suppressor genes, which normally act to inhibit cell division and prevent the formation of tumors. For example, the p53 gene is a critical tumor suppressor that activates DNA repair mechanisms and induces apoptosis in response to DNA damage. Mutations in the p53 gene can prevent these protective responses, allowing damaged cells to proliferate uncontrollably. Oncogenes are another class of genes involved in cancer. These are mutated forms of proto-oncogenes, which normally promote cell growth and division in a regulated manner. Understanding the mechanisms of cell cycle regulation and the factors that contribute to its deregulation is crucial for developing effective cancer treatments. In summary, cell division is a vital process for growth, repair, and reproduction. Mitosis and meiosis are the 2 primary forms of cell division, each serving distinct purposes. Mitosis produces genetically identical daughter cells for growth and repair, while meiosis generates genetically diverse gametes for sexual reproduction. The cell cycle is a carefully regulated series of events that ensure accurate and timely cell division. Mitosis consists of prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Meiosis introduces genetic variation through independent assortment and crossing over, which are essential for evolution and species adaptation. The regulation of cell division involves cyclins, CDKs, and checkpoints, ensuring precise control and preventing errors. Deregulation of cell division can lead to cancer, highlighting the importance of understanding these mechanisms for developing effective treatments. The study of cell division, its regulation, and its impact on health and disease is a cornerstone of biological research, offering potential avenues for medical advancements.